Welcome to today's episode where we unpack cellular processes of regeneration and aging. Our guest, Dr. Will Wang, develops spatial-omics and machine learning-driven approaches to decode how our muscle coordinates stem cell repair and how that coordination unravels in neuromuscular disease and aging. Join us as we explore how high-dimensional imaging, neural network analytics, and single-cell resolution maps are redefining our understanding of tissue regeneration and chronic degenerative pathologies.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Welcome to the Epigenetics Podcast. Thank you for taking the time to join me today. Before we dive into your research, I'd love to start with something a bit lighter. What's something that surprised you recently, inside or outside the lab?
Oh, wow. Surprised me recently.
Or if we rephrase the question, what did you do this morning?
Well, actually, I taught a course this morning. So one of the aspects of the lab is spatial biology. So I teach a course here for the graduate students on spatial biology, how to think about heterogeneity of tissue responses in terms of spatial localization within tissues and how that determines disease processes and how to understand their tissues a little bit better. So kind of delving into the heterogeneity of disease processes, especially when it's complicated. Yeah.
So do you feel that this is a new avenue that is like the future of maybe also epigenetics or science in general, that spatial is one of the avenues that it's moving forward?
Right. I mean, I think spatial has been a part of medical research for, 100 years or so, and thinking about anatomists and pathologists looking at tissues and how organization of tissue changes. I think it's been appreciated for all that time that diseases don't happen simultaneously throughout the body or any organ in particular. So, whenever you study a disease process, you really have to consider parts of the tissue and the parts that the signals are enriched in. So you look at kind of where biotech has been kind of pushing forward with the spatial technologies. It's really advancing over the last even 5-10 years.
That's true. So let's start or come to your scientific journey. How did you first become interested in science, and what led you to focus on the things you're interested today?
Yeah, I grew up, so my parents are kind of both. I grew up in a university. My mom taught physics and math and algebra, and my dad was working on the engineering side of things. And so I think I grew up in an environment that's very academically driven. And so when we moved to Canada and when I went through high school, science has always been something I was interested in. But biology in itself, I think, really was high school where I started really kind of enjoying learning about how cells work and how the body kind of works together. And at the same time, organic chemistry was really interesting to me at the time. And so when I went to university, I did biomedical science major specialization and got introduced to research at that point, where I think that trying to answer questions that people didn't have answers to, that really was exciting for me. And I love kind of problem solving and all the troubleshooting aspects of actually biological research.
So you knew from the beginning what you went into with doing an academic career? Because sometimes when
people... No, not at all, actually. I mean, I think my parents really wanted me to go to medical school, but I like the discovery aspect, I think, a lot more. And I have to say, I owe a lot to my mentors, my PhD mentor, my postdoc mentor, in guiding me through, and challenging me to do more and asking the hard questions. But
so how does this origin story still influence you today?
Of course, yeah. I like to push our research kind of in thinking about not just the low-hanging fruit, but what can we actually achieve with it, and how can we actually push the frontiers of how we study things. I like that aspect of thinking broadly as to where this research can go, and can we actually come up with both new tools, new assays, new ways of thinking about the biology that I think, we typically put on blinders for some areas of research, and you say, oh, that's too hard to do. But I think at times, if you can come up with a new method, you open up a whole new avenue of what you can discover.
So what is the limitation there? Is it that a method might not be yet available that you would need for this question to be answered, and then you would go and develop the method first, or would you what is your approach there?
Well, let me do an example from my PhD work. My PhD mentor, Dr. Michael Rudnicki in Ottawa, our lab really focused on how muscle stem cells and the genetic programs that underlie the stem cell activation and commitment processes, how that determines how the stem cell functions. And so in understanding how transcription factors dictate, and activate genes for the next part of the program. And so one of the major genes that he identified and the lab identified was PAX7, which is a master regulator of muscle stem cell specification. And PAX7 then turns on myogenic regulatory factors, including MIF5 and MyoD. And so MIF5 in itself, that activation process was really interesting to us because we can isolate muscle stem cells, and the majority of the muscle stem cells express MIF5 because PAX7 activates MIF5. But there was a subset of the stem cells that had PAX7, but the MIF5 wasn't on. And that subset of population was more stem-like and had better engraftment and therapeutic potential. And so in thinking about that, I went into his office and I said, hey, I kind of want to maybe do like a drug screen on these cells to see what turns on MIF5. What's the regulation of PAX7 that turns on its target genes? And the lab had actually several people working on that already. And so I thought maybe we can identify, extrinsic signaling pathways that regulated this process. And he said, that's almost too easy. Right? If you just culture the cells and you look for drugs that turn it on, that's not really complex. And we think that there's kind of like microenvironment regulators of this process. And so and he pushed me to do a screen that was in the stem cell niche. So without taking the stem cells out of their native environment. So I had to figure out how to culture, isolate and culture single muscle fibers with the stem cells still on them. And then identify and develop an assay to quantify how they divide, whether they turn on MIF5 or not. And that was not easy, but it took I think a few years to get that process up and running. But I think the reward is that this was something that no one else was doing.
Yeah. So I guess so you did your PhD at Ottawa Hospital Research Institute, as you just said, and this was one part. But I guess all of the publications that came out of this and the work that you were doing was revolving around this story, right? And creating supporting tools to then achieve this goal. Is this correct?
That's right. Yeah. One of the first publications that we that I had as a grad student. I was telling this story to another student earlier, that a postdoc in the lab had identified that the protein arginine methyltransferase CARM1 was methylating PAC7 in activating its ability to turn on MIF5. And he was a postdoc at the time. He had done this beautiful, chip and epigenetic analysis of that mechanism, doing overexpression, luciferase vector activation assays and whatnot. And then, because I had done all this work around MIF5 activation on single fibers, I started collaborating with him to then show that this process was actually happening. This interaction between CARM1 and PAC7 was occurring when the cells asymmetrically divided. And I think that kind of set up kind of a lot of the future work that I've been doing, in thinking about epigenetics. And a lot of other postdocs in the lab at the time were working on ChIP-seq and next generation sequencing. At that time, we were still calling it next generation sequencing.
Now it's just sequencing.
Now it's just sequencing, and thinking about how transcription factors bind to different parts of the genome, and then how that dictates how the cell behaves, right? And I started thinking about it in that cell identity and epigenetics in terms of what transcription factors are present in the cell and how they are regulated, right, in terms of turning on a specific gene program. And I think that's really interesting to think about from, at least our perspective of how stem cells in the body need to undergo several key transitions in order for tissues to repair, right? Muscle stem cells are kind of unique in this sense that they're really, really dormant cells. Muscle tissue actually is pretty robust and very capable of withstanding the stresses of life. So muscle tissue actually has a pretty low turnover rate.
But they also have like a very specific structure.
Yes.
To them.
Yeah. And so the stem cells in the muscle are actually mostly quiescent. They stay this very dormant state. And so it's a great model to study how stem cells maintain their quiescent state. But the quiescent state is also kind of maintained by the niche, right? The contact inhibition from the muscle fiber and its own unique kind of extracellular matrix that make up its basal lamina and its little microenvironment. And that kind of aspect was really interesting to think about. But then, when the muscle becomes injured, when there is an injury, the stem cell has to exit that dormant state
relatively quickly.
And then proliferate. So it changes from a G0 quiescent state, dormant state, to a rapidly proliferating metabolic state. And then going from that to then differentiation, which means cell cycle exit, cell-cell fusion for the muscle, and creating this new structure, which is a syncytia.
And they also need to move, right?
Yeah, they migrate. And all this process is really, there's a lot of outside-in signaling, but the cells have to change their transcriptional profile and their identity in this during this process. And so when I actually going back to the drug screen idea, when we first had this drug screen idea, we were thinking about characterizing the abundance of these cells, right? I was looking for MIF5 activation, and that's at the RNA level. So we could potentially look for an RNA signature for the process of MIF5 turning on. And we had carried out all these RNA, so FACS isolation of the different populations, and then RNA sequencing of the two populations to identify gene signatures for each. And it turned out, while I found, gene signatures for each population, as soon as the cell's activated, that signature changed. And that signature actually, this is occurring in the matter of hours, right? And so, and it kind of made me realize how dynamic the transcriptome is. And if the transcriptome is dynamic, then the epigenome has to be similarly dynamic.
Something upstream has to be dynamic. So before we move on, I want to put a timely context in this. So this was the work you just described was work that you did in your postdoc, right? Or
no, this is still in my PhD.
Okay.
Yeah.
So what kind of, if you say it's epigenetics, we end up in the territory of defining epigenetics. What are epigenetic factors? So what factors were you looking at that would influence this?
Well, so I think of epigenetics, and I think, depending on the person you talk to, the level of modification to the genome is going to be, slightly different, give you slightly different definitions. But to me, the epigenome is really about accessibility and the regulation that leads to different gene expression in each cell type, right? So it's kind of the identity of the cells. And whether it's modifications to the DNA, whether it's modifications to the histone, or it's simply transcription factor binding, those all contribute to this identity. And it's all, if we ignore the whole, somatic mutation that happen throughout the body, the concept that most of our cells share the same DNA, and the epigenome dictates how that DNA gets transcribed and into different identities of the cell types in the body, then transcription factors play a major role in this process. And so, to me, and from the stem cell perspective, and I did a lot of my training in developmental biology and cell identity, and the lineage specification and in turning on cell type specific genes, that's all dictated by transcription factors, bringing epigenetic factors that then regulate histone modifications and regulate the gene expression.
Yeah. So when you then did your postdoc with Helen Blau at Stanford, you characterized histone acetylation and also metabolic states, and this was done in quiescent activated and differentiating muscle stem cells. So the topic was the same, but you moved on to a different location and then continued your work, right? So
Right.
Can you talk about how you started this, how you maybe selected the lab that you were going to, and what you found?
Right. I mean, Helen is a pioneer in the field of reprogramming, right? And her early work demonstrated that you can fuse cells and overwhelm the identity of one cell with the cytoplasmic factors and nuclear factors from another cell. So her early work by fusing fibroblasts with mouse muscle cells in a specific ratio showed that you can turn on muscle-specific genes in the human fibroblast. And, these were very early evidence that you can actually change the identity of cells, even before the discovery of MyoD, which allowed us to do that with a single transcription factor. So her work and the lead up to understanding how stem cells reprogram during this activation differentiation process is important to think about. And so when I joined the lab, there was a graduate student in the lab, Nora, who's a co-first author on that paper. She had done all this beautiful characterization of muscle stem cell metabolism and how glucose metabolism actually regulated this acetylation process. And this was my kind of first foray into ATAC-Seq. And so when what she showed was that, like I said, when the stem cells activate from this dormant state, they have to become metabolically active, right? And a lot of that is using the glucose that's in their environment and metabolizing that for ATP use. But simultaneously, a lot of the metabolites from glycolysis are used. For example, acetyl-CoA is then funneled from the mitochondria to the nucleus, and that gives the acetyl groups for histone acetylation. What she found was that there were several critical processes where acetylation was generally inhibited, not by specific transcription factors, rather by the glycolysis program, right?
Okay.
So by modulating between the usage of acetyl-CoA in the TCA cycle versus lactate production, you can regulate how much acetyl-CoA is in the cell. And so at the moment of differentiation, it seemed that the muscle cells, the myoblasts, undergo this massive mitochondrial change, and that essentially shuts off the production of acetyl-CoA. And then you have to have, and the amount of acetylation in the cells just drop.
Okay.
And then what we ended up showing is that, by modulating that process, you can dictate whether the cells differentiate successfully or not. And so if you can boost the amount of acetylation, histone acetylation in the proliferating myoblast, you kind of reinforce that epigenetic state, the identity of the proliferating cell. And by doing that, they don't like to differentiate anymore.
Okay.
But during the differentiation process, if you remove the acetylation, then they differentiate better. And so it's like, at the time in the lab, we never made it into the paper, but we called it cellular amnesia, right? You erase the locations of acetylation in the genome because you just shut off the mitochondria, and you forget the state of proliferation because now you're not using those genes anymore. And you then turn on the muscle-specific genes that are required for differentiation. And so cell cycle is hundreds of genes, right? Requires hundreds of genes. So all that has to turn off before you can express the muscle-specific genes. And that kind of like provided a understanding of more global regulation of identity, not just transcription factor anymore, just, a metabolic regulation of global epigenetic kind of like determinants.
Would this also have applications for cancer? Because, they do the same thing. Not the same thing, but they proliferate. And if you shut off the mitochondria, they might stop eventually.
Well, a large part of that metabolic process is the lactate production in the Warburg effect of things. And one of the drugs that we actually used to modulate how much acetyl-CoA is being deposited onto the histone was DCA, dichloroacetate, which is actually known to inhibit cancer proliferation.
Yeah. There you go. So you follow this up by work that is now posted on bioRxiv since 2022. But I was not able to find it in a peer-reviewed paper yet. Maybe I missed it. So what you did there is multiplexed spatial proteomics and neural network analysis to resolve single-cell spatiotemporal atlas of 34 cell types during muscle regeneration and aging. I'm not sure if I want to ask this question, but is this paper close to being published?
This is something we've been working on for a little while. We started that project, and when spatial technologies were probably still very early stages, and we were developing tools needed to do multiplex imaging on muscle and the computational tools that are needed to identify all those different cell types, including rare stem cell populations. And looking at this in a spatial temporal way, we were able to, just take a piece of tissue, run this assay, and determine how well it's regenerating. I think that paper provided us with a lot of tools for subsequent papers that we've published in terms of identifying factors and spatial factors that were dictating how muscles regenerate versus how they age and whatnot. We have this paper is probably going to get incorporated into some novel findings that we're having, and so it's being revised and remodeled. But because, the spatial, I'll say the spatial kind of technology side has really advanced since we even pushed this paper out. And we really want to focus on biology that we've identified and things that we can apply towards actually modulating the activity of how muscle regenerates and how stem cells function.
So you're not only doing spatial work, but also multiomics. I don't know how this integrates, but you used multiomics to investigate the effect of prostaglandin E2 on muscle stem cell function. So what is multiomics in this sense? Because it can mean different things. And what did you find then in this study?
Right. So this was another project that the Helen's lab was working on when I first joined, and I think this whole aspect is really interesting to talk about. And so when I first joined Helen's lab in 2016, a couple of the postdocs were working on identifying factors that are necessary for stem cells to activate. It's like waking the stem cells up, right? And there, one of the first things that happens after an injury, a tissue injury, is inflammation, right? And
that's why the prostaglandin.
Exactly. And so the prostaglandins are secreted by inflammatory cell types that come into the muscle and probably other ones, and prostaglandins themselves kind of activate this signal process in stem cells in promoting their self-renewal and proliferation. And so Lenzon had published that, in HSCs, prostaglandins increase the self-renewal of hematopoietic stem cells, and that led to, translational attempts by Fate Therapeutics here in San Diego. And in other tissues as well, prostaglandin has been shown to promote stem cell proliferation. So, it turns out that the prostaglandin receptor is very highly expressed in muscle stem cells, and the postdocs in the lab at that time showed that prostaglandins were necessary for this activation process of muscle stem cells just by ablating the receptor of prostaglandin E2. So deleting that receptor in the stem cells don't proliferate, they don't activate, they don't really respond to injury, and the muscle doesn't go. It's not repaired very well, and in thinking about that, it's like it just makes a lot of sense. When there's an injury, you feel the pain, and then the stem cells activate in according to that, right? So at the time, we called this no pain, no gain in the lab. With exercise, you activate prostaglandins as well, and you feel the pain, and it's absolutely necessary for actually the stem cells to respond. And
so will they also lead to like muscle growth then?
Yes.
Or just repair? No.
Yeah. So the stem cells are needed for muscle repair, but we've shown in future publications, and at least in the aging context, that prostaglandins are necessary for actually restoring prostaglandin signaling is good for the muscle growth as well, hypertrophy. So that led to kind of the investigation in aging, right? Because muscles, as people get older, you lose your muscles. And you think about generally, my grandparents, old people that you see, this is a process that affects everyone, right? And so it's interesting and I think there's no real therapeutic option to boost your muscle for when you're
except for exercise.
Except for exercise, and exercise is not really an option for everyone, right? The big challenge, one of the big challenges for aging is that the muscles also don't regenerate very well. So if you think about somebody who's undergoing like a surgical intervention, and they're bedridden for X amount after hip surgery, for example, or knee surgery, the body's ability to repair that is reduced, and so it takes longer to recover from surgical surgery, and it takes longer to gain back the muscle mass that you've lost during that time where you're in bed rest. And so these are critical things that ends up having a very high healthcare burden, but how can we overcome it, right? And so one of the things that we thought at the time was that prostaglandin signaling was probably increased with age. People think about inflammation with aging and whatnot, but another postdoc in the lab, Adelaide Opala, at the time, we thought it was increasing, and then she did mass spectrometry, lipidomics, to actually measure prostaglandin levels, and it consistently was lower in the aged mouse. And so we thought, oh, fascinating, right? And so what she ended up showing was that there's this upregulation of the degradation enzyme for prostaglandin E2 and D2 called 15-PGDH, 15-hydroxy prostaglandin dehydrogenase. Now, and it seems to be pretty consistently upregulated in several aged tissues. And so what was kind of the amazing thing was that she did these experiments to inhibit this enzyme in the muscle using gene therapy like AAV or using a small molecule inhibitor, and the mice just got bigger muscles and stronger. It didn't work with young mice, so
Okay.
Unfortunately it's not going to work for everyone, but because the young mice didn't actually express this gene. Right, this 15-PGDH only became upregulated in aged muscle, and so when you target that in aged muscle, you restore the prostaglandin levels, and the muscle synthesizes new proteins, becomes bigger and stronger, and so we used our Codex method to detect where this protein was expressed, and we ended up finding that it was actually upregulated in the muscle cells themselves. This led to a subsequent study to figure out what was turning this gene on, and it turned out to be the innervation status of the muscle. And the muscle, as the skeletal muscle, is innervated, right? So you can control your movements. And when the muscle becomes denervated, it kind of has this epigenetic and transcriptional response that turns on a kind of developmental program that also mediates its atrophy. So it starts shrinking away when the muscle is not receiving any signal. So use it or lose it kind of.
Exactly.
And so we ended up finding out that if we just cut the sciatic nerve that innervates the lower leg, this 15-PGDH turns on quite rapidly. Actually, it takes about 7 days for it to turn on. So it's not necessarily rapidly, but it's very robustly upregulated. And this is concurrent to a bunch of transcriptional changes that occur inside the muscle. But in that case, if you inhibit this enzyme, increase prostaglandin levels, if there's a nerve injury, the nerve is actually regenerating faster and coming back to the muscle to reinnervate it. So this is one of the mechanisms as to why the muscle was functioning better after inhibiting 15-PGDH in aging as well. And so this prostaglandin signal was alerting cells to essentially damage. And if you dampen that signal, then it becomes kind of like non-responsive to just a worse response. And so the paper that you're referring to in Cell Stem Cell that we published last year was kind of is showing that the muscle stem cells themselves in aging, because there's less prostaglandins in the environment, that they don't respond very well to the injury. And by studying how this change, so we take the stem cells out, we give them prostaglandin E2, and you can see that the transcriptome becomes more normal, just like young.
Mm-hmm.
The cells reactivate their proliferative program. They don't die as frequently. One of the big phenotypes that we see with aged stem cells is that you isolate them, they die in culture. And so it's really hard to culture them. But by focusing our attention to the first, the early stages of stem cell activation, we can see by live cell imaging that they actually undergo this necroptotic kind of death. And so we profile the transcriptome, but then to really understand how this is working, how the drug is working, we decided to do a ATAC-Seq on the treated cells. Now we know that the response we've characterized the transcriptomic response at about 1 day after the activation process, but we really wanted to see if just this simple trigger can have a robust epigenetic effect very rapidly. I think we tested 2 hours after treating the cells with the drug, with prostaglandin E2. Mm-hmm. And we did a ATAC-Seq to analyze the different responses. And we had collaborated with Anshul Kundaji using their deep learning neural network model, which initially we tried BPNet, and then when they came out with Chrome BPNet, which is it's an upgraded neural network model, it's a convolutional neural network model that is able to separate kind of the profile of TM5 in ATAC-Seq from the actual transcription factor regulatory profile from chromatin accessibility. So the neural network kind of separates that and denoises the ATAC-Seq, but then learns the transcription factor logic within the cell and how it opens chromatin.
Oh, yeah.
It's really amazing tool.
So you don't need to do, because that would have been my next question, right? Is it enough to just do RNA-seq, look at the transcriptome, and then do ATAC to get the answers to all your questions? Because, the next step would then be look at, the binding pattern of a specific protein to chromatin, or to chip or cut and tag or something like that. But that would maybe do...
Well, yeah. So, I have to say, when I first ran these algorithms, I was absolutely blown away. So going back to the paper on glucose metabolism, regulation of chromatin accessibility. And we had done CHIP and we had done ATAC-Seq, and we can see CHIP for histone acetylation, right? And we had seen, just looking at the data, we can see that the ATAC-Seq can really pick up things that are correlated with the histone acetylation patterns. And the profiles of the ATAC-Seq peaks, because you can see kind of the transcription factor footprints, you can see, the nucleosome profiles are nearby. There's a lot of really rich information in that ATAC data. Now, if you're ambitious, you could try to go and look for patterns across these ATAC-Seq peaks. But the algorithms that we typically use are to pile up the ATAC-Seq peaks and look for enrichment of specific sequences, right? But that assumes that the sequences and the patterns are pretty uniform, right? But it's not necessarily the case. You can have, for example, this drug treatment, right? With prostaglandins, you can have multiple transcription factors that are dictating the epigenetic chromatin accessibility changes.
Yeah.
Right. So that would actually lower your enrichment score. If it's
you would see more motifs in your motif analysis,
right?
Then.
But if it's truly complex, then you become you dilute the enrichment scores, right? And what happens when you see multiple, you're not really certain which genes it applies to, right? And so what the neural network does, and it's amazing in my mind, is, we don't tell it what a transcription factor is. We don't tell it what sequences transcription factors bind to. You just show the neural network the genomic sequence and have it predict how much accessibility there will be.
Oh, so the other way around. So you don't show it the actual data, but just...
Yeah. And so over several rounds of training, it does a really good job of being able to predict, if you just show a genomic sequence for a specific cell type and condition, what the ATAC-Seq profile should look like. And then if you interrogate that neural network and then do kind of in silico mutagenesis of your genome, you can then and use figuring out the SHAP scores for each base pair. You can figure out that certain base pairs are really important for contributing accessibility at any given site. And so all of a sudden, you look at the base pairs with the highest importance scores, and it's a TF motif. Right? And for our cells, we can see the GC-rich E-boxes that MyoD and myogenic regulatory factors bind to. And so that was like, wow, that's amazing. And in the paper, we show some examples. For example, the PAC7 binding site that's very distally upstream of M5 that I studied during my PhD. It was able to identify the exact footprint of PAC7 bound to that cis-regulatory element, 57 kb upstream of the gene, and identified that a transcription factor is bound here, and this is the profile. These are the base pairs that it's binding to, right? So it doesn't tell you which particular transcription factor it is, but it does tell you which motif is important. And to me, all of a sudden, this combinatorial approach, because it doesn't just tell you one transcription factor, it tells you a bunch of them, right? All the different motifs that pop up, like MRFs, PAXs, homeoboxes, they all kind of show up upstream of any given gene that you're looking at. So the power of this, in thinking about transcription factor grammar and how it binds and how you turn on that gene is all of a sudden unlocked.
Mm.
Previously to this, I would have had to do, chip for each factor. And then you're reliant on antibodies. You're reliant on availability of antibodies, and sometimes they're not available. Sometimes they have off-target binding. And so this, to me, that was an impossible task before. And all of a sudden, ATAC-Seq neural network, this is a possibility to
Do you
even
then
think about.
compare the data that comes out of the neural network to your actual experiments?
Yeah. So
what does this then tell you? The comparison.
Yeah. So one, the neural network is able to pick up TF binding sites in some cases even more precisely than chip.
Mm.
So we compared that for CTCF chip that was available in our cell type. And then the other thing, I think this is more crucial, is that when we look at our cells that were treated with prostaglandin, we can then tell that the major effect is not just on MyoD binding sites that increase in accessibility, but also we had a whole bunch of sites that lost accessibility that were associated with AP1, but not just AP1 and other binding partners, including AP1 and CTCF sites, AP1 and NFI sites. And I think that tells you that AP1 is kind of mediating a couple of other transcription factors in this aging process. And those can be followed up with in future experiments. But you can now see that it's not just necessarily one transcription factor that's changing, but it's the transcription factor and how it mediates other transcription factors and recruits them to all these different sites that happen. And to me, this is really interesting to think about because, we show with prostaglandin E2, but this could be applied to any drug treatment. Mm-hmm. How does a drug change the transcription factor profile of a cell?
Yeah. Very powerful, I guess. So, in interest of time, because it's already 47 minutes into the interview. I forgot the time. Since 2022, you now run your own lab at Sanford Burnham Prebys, and this is a question I always ask in the beginning, but now we skipped over this. Could you maybe introduce your current role that you in now?
Yeah. So I'm an assistant professor here at Sanford Burnham Prebys. My lab focuses on neuromuscular conditions and how interactions between different cell types, including stem cells and inflammatory cell types and muscle and nerve, actually dictate the progression of the disease. And also we use a lot of spatial and multi-omic approaches to understand what goes wrong in complex diseases. And it's amazing because, muscle in itself is quite a unique cell type. It's multinucleated. It has, unique domains where the nuclei, even though it's a syncytia, some of the nuclei have a totally different epigenetic program than nearby nuclei. So the ones that are underneath the nerve express different genes and have different transcription factors compared to the other ones that are kind of scattered throughout the muscle. And it's really interesting to think about how you have different transcriptional profile within one cell and how those, how spatially they're determined and specified.
So the lab is active now like 2 to 3 years.
That's right.
So I guess it must be running smoothly now. So is there already some results that you could share?
So the lab is kind of focused on, we like to, like I said earlier, have some technology development, and then also have some, more validation type of experiments delving into mechanisms of disease, right? And they work with each other in terms of how we push the biology forward. So we've developed. We're continuing a lot of work on multiplex immunofluorescence and looking at 50 to 60 antibodies or different staining patterns in the tissue. And we've developed a couple of panels of reagents to look at specific changes in the post-translational modification of proteins. So one panel is specifically looking at glycosylation within the tissue, and the other panel we've actually collaborated with Active Motif on looking at epigenetic factors, including histone modifications, different compartments, markers of different compartments, DNA damage, etc. And that's actually revealing some very interesting insights into, how global changes occur for things like histone acetylation across different cell types and different cell states.
Mm.
And we see the same different increase in, for example, H3K27 acetylation in proliferating B cells in the spleen compared to more immature versus differentiated B cells that have less histone acetylation, very similar to the muscle stem cells.
Mm-hmm.
It's kind of amazing to us that going from cells in a dish to then tissues, right? And how diverse your epigenetic like modifications are within the tissue. By looking at the combination of some of these epigenetic modifications, you can tell different cell types apart.
Of course, yes.
But not at specific sites across the genome, right? But just the
Globally,
abundance
yeah.
of those marks.
So you would just have like one intensity score?
Yeah.
Yeah.
Per
mark. Yeah.
Per marker, yeah.
Yeah, I mean, this comes back to the chrome-HMM, right? To see the different chromatin, domains, and then each cell type should have their own.
Right, exactly. And metabolic states and how that kind of are co-regulated.
So for the last, let me see, 52 minutes, we've been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I think what I'm mostly interested in is really this idea of how cells change their identity, especially stem cells in their activation and differentiation. You go from the idea of reprogramming your identity from a quiescent state to a proliferative state to a differentiated state. But that also can be harnessed for, regenerating the tissue, right? And then also for rejuvenating an aged organ. And I think, to me, we're at this stage where we can really take our epigenetic kind of understandings and start applying it towards, treatment, enhancing the body's ability to repair itself, or even restoring the capacity of aged tissues or diseased tissues to restore their normal function. I think, to me, that's where we're headed.
Yeah, I guess that's also a big opportunity, right? Because if people are getting older, they get injured, they get those surgeries and everything. So if you can help them regenerate faster, you save a lot of money as well.
Exactly. Yeah. And the population across developed countries are all getting older, right? And so this is why aging research is going to play such a major role in the next couple of decades and how it shapes, healthcare for the future.
So thank you, Will, for your time and for being on the show.
Thank you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.